New coronavirus variants have been detected in over 40 countries, including the UK, South Africa, Nigeria, and Brazil, where they first emerged.
Now health officials say a new threat is identified in South Africa.
British officials imposed a new lockdown across England.
Recent reports of a new, highly contagious coronavirus variant in the United States.
In the US, multiple states have reported new variants, or versions of the virus with mutations that could change how it spreads. New research suggests that some of these variants are highly transmissible, and at least one may reduce how well vaccines work. With countries around the world already facing escalating infections and overwhelmed hospitals, these mutations could make ending the pandemic more difficult. We'll explain. Like other viruses, the coronavirus spreads by infecting cells and then reproducing within them, creating copies of itself that spread throughout the body. As new versions are made, the virus's genetic code, which holds the instructions for building new virus particles, is copied over and over again. But the code isn't always reproduced correctly. Small genetic mistakes, or mutations, can create new viral variants. If the new version of the virus has mutations that allow it to spread faster than the current most common variant, it could lead to a surge in new cases. This is already happening in places like the UK and South Africa, where some of the newest ones were first found. Current data suggests that the UK variant could be 50% to 70% more transmissible, but the research is still early. To determine how transmissible and dangerous a virus mutation might be, scientists run a series of experiments in the lab. The first step is genetic sequencing.
The genetic sequencing gets to the blueprint of what the viruses are. We can use samples and data from actual humans to figure out what the genetics of the viruses are.
To do this, scientists take a sample of the mutated virus and sequence its genes. Then, computational programs help scientists identify mutations.
Once we know what they are, and now we can see that, this is an important population, we can start manipulating the virus. We can either start working with clinical isolates of that virus, or we can actually make the strains. So putting those individual mutations into the backbone of viruses that we already have.
After sequencing the virus, scientists test the variant in a lab.
What we do is we compare how well the virus replicates in different types of cells. And so there are two ways. We will just do infections and kind of measure the level of the virus over time in these cell types. And the other thing that we'll do is a competition assay, where we'll take 50% of the wild type, the original strain, and 50% of the mutant strain, and then compare how well they grow. If there's an advantage, we'll see that after 24 hours or after 48 hours, one of the two might outcompete.
During lab experiments, researchers can also test whether human antibodies created by infection or vaccines are effective at fighting the new form of the virus. After culture testing, scientists typically move on to animal models, and then sometimes to humans. Right now, the research is still early, but scientists are beginning to get a clearer picture of what these variants look like. From sequencing, they know the UK variant has 17 key mutations.
These variants that we're seeing, these changes, give a small advantage in terms of the ability of the virus to replicate or transmit.
This has a lot to do with the spike protein, which is critical to how the virus spreads. A portion of the spike binds to a receptor on human cells, like a key fitting into a lock. This enables it to enter and infect. Scientists found that the UK variant spike proteins have 8 amino acid changes. Previous research appears to show that some of these changes may be increasing the protein's ability to cling onto and enter human cells. This could be making it more transmissible, which means a higher vaccination rate may be needed to reach herd immunity. Another big question is the virulence, or how harmful these new versions of the virus could be. Scientists say that most likely, the new variants don't cause a more severe form of COVID-19. Another question is whether the new versions will show up on tests. Early data appears to show that most COVID and antibody testing will continue to work.
We should still be able to tell if you have COVID. What's less clear is if we can tell if you have a specific variant or not.
The most pressing issue is whether vaccines will still be effective. For the UK variant, it appears that they will. However, for the variant originating in South Africa, preliminary data is worrying some scientists. Researchers say that any of the more transmissible variants could eventually become dominant.
We've seen variants take over in terms of the D614G mutation, and it wouldn't surprise me if the South African or the UK variant do a similar thing where it becomes the dominant variant around the world just because of a small fitness advantage or small changes that make the virus more fit.
To try to stem the spread, certain countries like the UK have already imposed strict lockdown measures.
The number of patients in hospitals in England is now 40% higher than the first peak in April. It is inescapable that the facts are changing and we must change our response. And so we have no choice but to return to a national lockdown.
While the new variants are concerning, experts say that current prevention measures should continue to work.
This isn't something that's completely overcoming masks or distancing. And so if these measures are put in place in more strict or more effective ways, it'll be just as effective, if not more effective. And the outcome may be that, being a little bit more serious about this will get transmission as a whole to be reduced. And I think that's an ideal.
Scientists say the coronavirus pandemic could be on the cusp of a major shift, but more research is needed to know how severe the impact of the new variants will be.